Hi, now we're at lesson C of part one of machine learning and cloud services. It's April 2019, and here's Jeffrey Fox extolling the virtues of clustering and WebPlotViz, which is used to visualize clustering, in particular k-means clustering. And here we have a couple of examples, and we will now actually on the next slide see some actual further examples of WebPlotViz. So let's go and do that now. All right, here we are. We're using WebPlotViz. We go to the so-called public dashboard, which you can get to from the spider.gwdsc.soic.indiana.edu link. And you will then find at the public dashboard some collections. If you're in E222, you go to the E22 collection, and you will find it here at the top of the second group, and you press view. Then you will see that this collection has 5 plots in it. Let's look at this one here, one of the a group of 1,000 dating related points clustered from the book by Harrington, and you can see here it is. We can actually now rotate it. These are three-dimensional points viewed in 3 dimensions, and you can see it's got reasonable clustering. It's not unique clustering. It's a long thin white with a bit of an extension here, clean red cluster, and a relatively clean, somewhat broader green cluster, and together they divide the system up. So we can make the system smaller, bigger, and we can change the size of the so-called glyphs, except these are points, not glyphs, so we'll change the size of the points. There we make that just, all I've done is change the points from being large to small, and maybe sometimes that tells us something. In this case here, it doesn't make so much difference. All right, here we have a rather old piece of work of mine, which I always like, because if you look at this visualization, which is 96,000 proteomic points, they have the points basically are shaped like a handbag, very rather clean handbag, with these funny, I've never seen another set of points look like this, with all with such a clear overall shape, with nothing in this region here of the handbag between the two handles. So if you look at this data, the colors are not clusters found by some magic program, but just biologically related proteomic sequences identified by the experimentalists. So you can already tell a lot from this visualization. Let's look at these red points over here. They're actually pretty near each other. And if you look in 3 dimensions, you can really see they're pretty clustered. There are a few outliers, which suggest maybe there was someone misclassified, but that red group is pretty much all there. Notice it doesn't show up as a separate cluster in terms of the points. It just there is a region of this handbag, and there the red people live. And there is another set of blue people here. If we look at the blue people, you can see they're sort of near each other, but they're much more scattered. They're all over the place here. And so it's not clear to me whether those really are associated, or whether the blue people on the left really are sort of different from the blue people on the bottom center and this little blue cluster over at the top right. It's not clear to me. If we look in the middle, we have actually, this is where the biologists were focused. We actually have a set of several biological families, and the biologists were happy because these biology sets were actually meant to be similar to each other. So the yellow and the reddish and the pink were meant to be similar to each other, and they are similar to each other. So you can see this plot has shown you a lot. It's shown there are some successes, but also that you're not going to find those successes by clustering, because they're just not distinct as physical clusters separated in this particular space. This space is defined by the so-called Needleman-Munch distance, which is a variant of the Smith-Waterman. So that's that example finished. Okay, here we have as our third example on another set of fungi. Well, actually, it's a set of fungi. I've done lots of work with fungi, which we will see other examples from screenshots later on. But this is pretty interesting. 446,000. They're clear clusters, but these clusters are sort of strange. They have funny shapes. Like here's this white cluster. These actually all look like tadpoles, long, thin tails. I think the long, thin tails are not coming from Mother Nature. They're coming from the messed up distance measures. But I've never fully understood or proved that. There's some structure here with lots of related long, thin things here in this white group here. What does that mean? And you'll find that pretty common. You often get lots of separated sets. What that means, I don't quite know. So if we look at this, we have clearly separated things. These white ones over here are separated. Green one here is separated, but in this middle here we have a complete mess. And the clustering program, which is a variant of k-means, which works in the case where you have just distances. I say this is actually Smith-Waterman distance, not Needleman-Munch. Needleman-Munch doesn't work for this type of sequence, which we found out to our cost. But you can see these clusters are not well separated. There are lots of close together things, and it's not totally clear, except on the size type issue, that they are separate clusters. Here the clusters are determined by size, approximate size. And so this is very rich, and so we found lots. So you can say there are clearly lots of clusters here, which are species. Exactly the quality of the separation between the species varies across the set of points, set of DNA sequences. And there's a whole richness which you can explore by better distance measures. I think you can do actually better clustering. I think given the distance, this clustering is about as good as you can do. You obviously can do more experiments with more points and so on. So it's sort of pretty interesting, and sort of illustrated the depth that you can get to in this type of problem. Okay, here's our last example. This is a set of points not clustered but actually labeled by the biologists, and this is actually pretty neat. They're very clean. This is a much nicer data set than some of these other ones. Maybe the data was higher quality. There's still a little bit of distortion, but some, like this nice red cluster, is so beautiful. There are lots of points in it, and they're very clear. They're separated. There's almost no noise. If you look at noise, you can see sort of the noise is actually clustered, and so it may just be families off in left field. Like over here, we have this light blue cluster here. We have a much stronger brown cluster here, and then over here we have some interesting structure. We've got a long blue. Maybe that is actually two clusters not properly separated, and the same is seen over here, where obviously it looks as though there's a richer structure than the biologists have identified. There are three bright green clusters here which are obviously separated, and and you can tell that some of these things which look on top of each other, when you rotate them, they become separate. Like this red and purple thing looked on top of each other, but when you rotate, you'll see they're clearly separated. And you can see also they fall, in this area, they fall into groups. There's a big group over here, a group here, and a group here, a little bit of scatter, isolated cluster here, isolated huge cluster here, and so on. So this is really shows the power of these methods. You really can actually tell what's right, what's wrong, and that you have a great confidence in what you're going to do going forward with visualizations like this. Right? So if you look at the raw data, I challenge you to have the same confidence. All right, you just saw some examples of WebPlotViz. WebPlotViz is a program we wrote in my research group, 10,000 lines, largely JavaScript. And the reason it's JavaScript is a client-based system running using the HTML5 specification, using a code called 3.js, which is a library. And if we want to, what we're doing here is usually called dimension reduction, which is when you do data analytics, you have a set of points, which typically are either in a large dimensional space. Sometimes they're in 2 or 3 dimensional spaces, but more often than not, they are actually in a large dimensional space. And then you have to look at them. You can't visualize a 93-dimensional space, so you have to reduce it to 3-dimensional space. Alternatively, as in those DNA or proteomic sequences, you don't actually have a vector space defined. You just have distances. There are no scalar products. It's not a Euclidean space. They're just distances. And you need to have algorithms that take points where you only define distances and map them into 3 dimensions. That is done by a technology which tries to keep the distance in 3 dimensions between any 2 points similar to the distance very close to the distance in the original space, where either you only have distance or you have actually a vector space. The 2 famous methods here are principal component analysis, which you'll find in almost all libraries, which is a linear approach. It's very powerful, possibly the one that's most often used. And I don't go through it in these lectures because it requires knowing about eigenvalues and eigenfunctions, eigenvalues and eigenvectors, which are out of scope for these lectures. And then the method that we usually, which we use for the data you just saw, we use so-called MDS, multidimensional scaling, which is a nonlinear transformation, which is a complex optimization problem. And they all map abstract spaces to 3 dimensions. They all run well in parallel, and they give, as far as I can see, great results, and more people should use them. And to visualize, we used to have a package called WebPlotViz written in Windows technology, but we switched to this browser-based technology over several years ago. And here is a specification of it. Here are some examples. We'll see these pictures later on, these typical examples. And here we have a typical screenshot, and it shows rather more than what I showed. It actually shows you how, in this thing here, more information about the actual, the play, pause. This particular program actually visualizes time series, and so that's why you can have it's useful to have a pause, because you might be running it forward in time. We applied this to the stock market, and then it also they have all these clusters, or again, in this case here, clusters are not found by clustering programs, they're just labels. Like you, all stocks from the energy sector are colored green, for example. And you can change colors and remove clusters and things like that. And that's what these controls over here are describing. So it's visualizes 3D plot sets for either fixed plots or for time series, which are just sequences of 3D points, which are ordered in time, and then in the case of time series, it just displays them as a function of time. Data management is done with a simple client interface, and it's all stored on the back end in MongoDB, which is one of the most famous cloud databases, NoSQL databases. There is 3.js, WebGL, JSON, Play Server framework, and that's what we have a paper which is referenced on this website, which describes it in more detail. So here's as well as the two-dimensional examples, the static examples I just showed, where fixed set of 3D points. We can say have this visualization. In the stock market, there are around 7,000 distinct stocks. So at any one time, you have 7,000 points, which again, we reduce those points from whatever dimension, whatever abstract space that we looked at, which was, as we looked at just the values as a function of time to give them a position in space. And we can look at them from the date when we did this at almost 3,000 distinct times. And say the labeling was defined by various Wall Street and Dow Jones classifications. And the data comes from the Wharton Research Data Services, which is available to university students for research. And we went from January 2001 to December 2015. And we use this, which basically date, symbol, price, and various corrections that they do splits and things like that. Okay, this is the streaming stock example, and we'll start it. This has a play symbol, and it has this has lots of features, and one feature it has is to actually form trajectories corresponding to particular stocks. We have let's actually, we're a bit off. Let's change the scale here. This trajectory here is Apple, and somewhere inside here we have British Petroleum, BP. I chose to do two trajectories, BP and Apple. Apple is the most energetic of one of the more energetic stocks, tending to increase, and BP is less exciting, screwing around in the middle. In this time period, oil stocks didn't do so well, at least after the crash and things in 2008. So you can see these are crazy trajectories. And this, we'll just let this thing run. You can cancel it if you get bored. You can see, what do we have over here? We have actually displayed the position of these are trajectories. So this is this has today's position and also the previous positions of previous years. And these things like finance, which is the greenish cone, it's actually just the current value of finance, which actually you can just see over here. There's finance around here. Again, we haven't got the colors quite so distinct. Health is an orangey cone, and these pseudo circles are actually positions they would have if they increased flat with year 10% or 20%. And so we have technology, energy, industrials, consumers, finance, real estate. These are all the classic Wall Street classifications. I think they're the top 10 stocks of each of those categories. The overall background is the 7,000 stocks colored in a way which you can't see because I have them too small, in the way that reflects the amount of change they've done. So now we have so here we have Apple really making noise here. So, while this thing is playing, we can rotate it to see we understand what's going on. As you see, this is pretty complicated, and that's one of the problems we found in this visualization. It was sort of a bit too complicated. It shows some interesting structure, which we'll see some screenshots later on. But it certainly wasn't drastic. There wasn't clean structure, which of course is you'd expect. If there was clean structure, everybody would be a billionaire by exploiting that structure. The fact that the structure has huge randomness, or not randomness, uncertainty and unpredictability, because you can't really tell whether when A declares war on B, or when there's a typhoon or a disaster of various types, you just don't know what's going on. So this yellow is the 20% number. And the lighter yellow is the 10% number. Here we have Apple doing pretty well. But even Apple doesn't have a smooth trajectory. It's pretty difficult. You can make it bigger, of course, to see the structure. And everything is now, in the stock market, things average down, and so now they're all in the middle. These stars, what are stars? Well, not certain what these green stars. Energy. They're energy stocks. All right, we're nearing the end here. That's it, it's finished. December 2015. All right, so there's lots of ways to study this and get rich. So that's the end of this this particular view. And here is just a list of some resources. The resources exist on Cloud Mesh, on the on the GitHub for the class, and it has so here's the online URL. Here is a YouTube video describing it, which is pointed to on this URL, and you can create your own account. You can upload a plot file, and you have to have index X, Y, Z, cluster ID, and label. And if you don't have a label, you better just use your, you just put a number like 0 or 22. There was a more powerful syntax defined in XML for more complicated cases, but that's not necessary for simple cases. Now we just finished this slide set with a set of resources. So here is the collection of the WebPlotViz resources, the k-means examples, all defined in GitHub. Here is a place where we have the things like the scikit-learn versions of k-means. As you'll see in this slide set, we have various examples like t-shirts and dating. They're available at these links. And this goes in a little more detail. There is a Python program which I use to generate some artificial clusters, 4 clusters in 2D, and to illustrate what happened when you kept on running k-means and sometimes got the wrong answer. There's also parallel code, which I discuss at the end of this unit. And this is the t-shirt example, 85 points. And here is the markdown, the basic GitHub documentation. And here is the scikit-learn documentation. And here is the KNN material, which is actually that's in the third part of this lecture sequence. No, it's not a KNN is related to clustering, but we describe it separately in the third part of this unit. Thank you very much. That's the